All right, guys. How y'all doing? As you log in, please look at the screen and you'll see there's a poll there ready for you. I want you guys to start from scratch. Draw and label a nephron using a scratch piece of paper. If you're already done, probably logged in a couple minutes early, but I want you guys to completely label that nephron. So as you log in, hopefully you'll see this poll. You guys can click done when you're done. Or, like Heather did, you can click that Dr. H is so mean. All right, for those that are logging in, make sure you're drawing and labeling a nephron. That's you guys' goal. Try to include everything that you can think of. You could use abbreviations. The abbreviations definitely speed things up. All right, we're getting closer to people getting finished. For those that are just logging in, you're drawing a nephron from scratch and you're labeling everything that you can possibly think of. If you leave something out, that'll be okay. I highly recommend trying not to use your notes. Go as best from memory as possible, because who cares if you get something wrong? That's what this is all about, right? It's about learning. Can't be so worried about perfection the first time around, in my opinion. Melissa is joining in on the Dr. H is so mean. I'm so cruel. Do it just to see who falls for my trick. Instant F. Too late. Yeah, Tariq. I've seen your car, man. You'll just drive around traffic. You'll like actually make the wings go out. You'll fly over traffic. Nice. All right, still quite a few people that are working on it. Becca, Alyssa, Veronica, and Tariq, do your best. You can draw it out as fast as you can. Trying to give you guys that practice, that well-needed push, just like an exam would. It's going to push you to make sure that you, are doing things in a timely manner. If you haven't studied it since yesterday, now's your chance. Throw it out there. If you remember absolutely nothing from yesterday, then that means that you need to pay a little bit more attention during lecture, which is sometimes hard to do, I know. I'm going to go just one more minute, and then we're going to call it a ball game and get working on it. Remember, I'm just asking you to draw the nephron. Oh, yeah, that's a super cool pick. Surface electron microscopy stuff is super cool. Alyssa, you're working on it, I'm assuming? And I know, Tariq, you're probably working on it too, I'm assuming? All right. All right. I'm going to go ahead and stop this poll and share my screen. Just, there are a lot of ways you can do this fast. You guys need to work on speed, because your medical programs are going to require a lot of speed to get through stuff. Smart, efficient, concise, speed. So when you guys go to draw it out, just don't worry about perfect lines. I never do. It's all about labeling. It's all about understanding what you understand. So blood enters through that afferent arteriole, goes into the glomerulus. And the glomerulus is a fenestrated capillary. It's a modified capillary bed. It's got fenestrations, lots and lots of holes. The afferent arteriole is bigger than the efferent arteriole, so the pressure builds up in here, and that's going to be one of the primary drivers that allows for filtration. So now all this fluid gets filtered out into that Bowman's capsule area. Doesn't have anywhere to go from there. So where's that fluid going to go? Where's that filtrate going to go? It's going to enter the PCT. It's the only place it can go at this point. We're going to talk about percentages and some math and stuff like that today in terms of how this stuff is getting filtered and how much gets filtered. But it's about 20%. 20% of the plasma that enters your kidneys is going to get filtered. That means that 80% is going to still be there. We're talking about 80% plasma volume. So 80% of your plasma volume is still there as it leaves. So 80% is going to go in that efferent arteriole. So let's follow the filtrate. The filtrate now goes into that PCT, the proximal convoluted tubule. And that thing's twisty, windy, crazy, convoluted. But it will eventually descend down into the renal pyramids. And the anatomical portion of the nephron that descends into those pyramids is called the loop of Henle, or the nephron loop. And there's a descending limb and an ascending limb, and there's also a thick portion and a thin portion. Structure determines function, so making sure that you understand the differences in each one of those will be important to your education. So stick with it. From the ascending limb of the loop of Henle, it's now going to pass up into the distal convoluted tubule. The distal convoluted tubule, it's only called distal because it's not attached to the glomerulus. So we're talking about the filtrate, really. The tubule itself is actually winding right back up next to this glomerulus, right? We only draw it out linearly because it's easier to do so. But that DCT comes back into contact with the glomerulus from whence it came. This is the juxtaglomerular apparatus. Did everybody that drew their nephron add the JGA? Just be honest, yes or no. I'm guessing some of you forgot it, and that's okay. No worries, Beck. Thanks for your honesty there. Just Sabrina, Veronica, just try to remember that that will be an important part of, cool, Andy. That will be an important part of this whole nephron. And it's definitely going to be a major player in the physiology of how it goes. So that's basically, a portion of the distal convoluted tubule here, boom, that's the JGA. So it actually flips back. The distal convoluted tubule will eventually drain into the collecting duct, the CD, and it's going to pass through these little collecting tubules to enter the collecting duct. The collecting duct is eventually going to lead to the minor calyx through the renal papilla, at which point we have urine. Anything up to that point is not urine. It's still called filtrate. Filtrate, 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 filtrate through that whole system is filtrate. It's only urine once it enters the minor calyx. Is that clear? That's an important part of this, because what that means, if it is still filtrate, then it's still going through a process of the nephron. Let's talk about these processes of the nephron, and then we'll come back to our blood supply, because that's important as well. But the first process occurs, boom, right here between the glomerulus and the Bowman's capsule, because there's a high amount of pressure, and because of the fenestrations, and because of a number of other properties that we're going to be discussing today, we have massive amounts of filtration. How much of our filtration in our kidneys occur between the glomerulus and the Bowman's capsule? What percentage of our filtration occurs here? What percentage of our filtration occurs here? Not what percentage of the volume of plasma that enters gets filtered. I'm asking, of the filtration that occurs in the kidneys, how much of it occurs here? The answer is 100%. 100% of all filtration occurs here. There's no filtration out in the PCT. There's no filtration in the DCT, loop of Henle, nowhere else. All filtration occurs here. So we're going to be talking about the properties of the glomerulus, the properties of the Bowman's capsule that allow for filtration to occur there. Once that filtrate is filtered into the Bowman's capsule, now it enters the PCT, at which point the PCT is a big old fuzzy teddy bear, meaning it's got a lot of surface area, right? And that surface area is going to allow for the material, massive amounts of material, 180 liters per day that it's getting pushed into your PCTs. Well, the large majority of that right here in the PCT, somewhere between 60 to 80%, it's a big number, is going to immediately go from these tubules and it's going to go back into the bloodstream. This is called reabsorption. Okay, where does the large majority of reabsorption occur? The PCT, the loop of Henle, or the DCT? Or the collecting duct, for that matter? Good, 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 good. Just trying to do that whole redundancy thing. No, 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 not 100% of the blood. Not 100% of the blood. 100% of the volume that gets filtered. There's a change in terminology. 20% of your blood gets filtered. So that 20% now becomes 100% because that 20% is filtration. It's filtrate. Does that make sense? 100% of filtrate and 100% of filtration occurs in the glomerulus. I'm not asking you in that particular question what percent of your blood gets filtered. That's a whole nother ball of wax. I don't even know if that's the right saying. Ball of wax? What would that even mean? Who knows? Yep, 20%, but that's even different, Melissa, that's even different, okay? 20% of your blood volume goes to your kidneys. Of that 20%, 20% will be filtered every minute. And of the amount that's filtered, only the plasma matters. So we're going to come back to that. The length of the tubules, I love it. Yes, okay. So the longer the tubules, the more transporters. The more surface area, the more transporters. So absolutely, there is going to be a relationship between the length of the tubes and the amount that gets reabsorbed. Not the amount that gets filtered, though. So we're going to come back to that. The loop of Henle is a great example of what's going on there. Are there any more questions about that? Because we're going to get it moving. Got it. Awesome. So here we are in the PCT, we're reabsorbing. I'm going to walk you guys through later about the sorts of materials that are getting reabsorbed. But ions, bicarbs, glucose, 100% of glucose gets reabsorbed here. And where does water want to be, y'all? At the party. And as long as the membrane is permeable to water. So here in the tubule system, there's no cracks. It's not how it works. Out here in the capillaries, there's cracks for water to be able to push through. But this membrane is impermeable to water unless there are transporters for that water. Those transporters are called aquaporins, so we're going to be talking about them later. But here in the PCT, there are aquaporins. Therefore, as ions, such as sodium, potassium, calcium, chloride, as they're getting reabsorbed here, bicarbs, phosphates, as they're getting reabsorbed, glucose, water follows. Whereas down here, things are going to change. Structure determines function. Structure determines function. I can't say that enough. What do we know is allowing for reabsorption? Protein transporters. So as we go through this system, the protein transporters are going to change. In fact, as we go down into the thin descending limb of the loop of Henle, there are no protein transporters for any ions, any glucose, you name it. The only thing that's going to be allowed to be reabsorbed here is water. So in this thin portion right here, this is a reabsorption, but it's water only. So in the thin portion of the loop of Henle, only water can be reabsorbed. So just because this is what I do, what's the only thing that gets reabsorbed in the thin loop of Henle? Both descending and ascending, by the way. Oh no, I dropped my pen. Water. Now where do we know water wants to be? Where the party's at. So we're going to have to figure out how we get the party out here in the blood. That's going to be a whole nother thing. But we need the party out here to attract that water. So we're going to talk more about that probably tomorrow. I'm not sure we'll get to it today, but we'll try. We can kind of throw it into the next portion of this. The ascending limb, as it gets thick again, the thick ascending limb of the loop of Henle, the transporters change. The whole entire epithelial tissue starts to change here. We start not having that big surface area, all those microvilli, they just don't exist here. And the types of transporters are going to change. We're not going to have transporters for glucose. We're not going to have transporters for bicarb. Instead, there's going to be one major transporter found here. There are other transporters, but we're going to focus on one. You guys only have to learn one transporter that lives here. That's it. You guys have already learned it. This transporter is add sodium, 2-chloride, potassium, Symporter. You guys remember this one from the digestive system? So if we have this transporter in this cell membrane here, what's being reabsorbed in the thick ascending limb of the loop of Henle? What's the primary thing being reabsorbed here? No, not bicarb. I see why you said that, because we were dealing with the chloride and stuff like that, Ruju, in the digestive system. But here, don't overthink this. What transporter is found here? Sodium 2-chloride potassium. So what's going to be reabsorbed here? Sodium, chloride, and potassium. Nice and simple. Sound good? And that reabsorption is occurring from the tubule system, and it goes into the blood. So now you might notice that I've put in the capillary system here. If I have a capillary system out in the renal cortex, what capillary system are we dealing with here? What capillary system is out here around the PCT and the DCT? You can do it surrounding the tubes. So this is the peritubular capillaries. Ring a bell? Whereas down here is going to be the capillary system that surrounds the loop of Henle. So what are we dealing with down here? Vasa recta. Right, so we got the vasa recta down here. And what I want you guys to notice is the direction of the blood flow that I have drawn in. You guys see how I have the paratubular capillaries? They're wrapped around the PCT and the DCT, but the blood is flowing across those and then diving down. And the vasa recta is now going to have its blood flow going in the opposite direction. This is called counter current. So the fact that this current is going in the opposite direction, this is called counter current. Without going into any detail right now, I'm just doing this. Yeah, it's just following the capillary. The capillary just flows in, you know, the capillary, just imagine one big long tube, a garden hose, but it wraps back around the other way. So you got a garden hose that's leading out towards your your plants, but you know, you decide to plant something back on the other direction. So you're going to have water flowing through the tube one way, and then you've wrapped that hose back the other way. So now the water's coming back the other way. Okay, why is this important? What's being reabsorbed in the thick ascending limb of the loop of Henle? Sodium, chloride, and potassium. So what's happening to the blood concentration of sodium, chloride, and potassium right here in the vasa recta? What's happening to the sodium, chloride, potassium concentration? Increase, decrease, no change inside the vasa recta. And where does water want to be? So now as this blood is flowing back towards the descending limb of the loop of Henle, and it's going to be passing these thin portions here, the water moves right back towards that blood because that's where the party's at. Does that make sense? This is the counter current. The counter current allows that to exist. Without the counter current, it wouldn't work. DCT, CT, and CD. These are all unique. These membranes have all changed yet again because that's what they do, right? Structure determines function. So out here in the DCT, the collecting tubule and collecting duct, Heather. Out here in the DCT, CT, and CD, we have a whole bunch of cells, like intercalated cells and principal cells. And these cells don't, what's the best way to put it? These cells are just dependent upon hormones. I'm just going to be as straightforward as I possibly can. These cells are dependent upon the hormones that are in the system. The hormones that are in the system are going to determine how many protein transporters are found in these cell membranes. So the DCT, the collecting tubules, and the collecting ducts, they don't have permanent residence. They do not have permanent transporters. Does that make sense? Their transporters, their up and down regulation of their transporters is dependent on which hormones are in the system. Also dependent on things like hydrogen concentrations of your blood. Let's put this into perspective. I'm just going to write hormone dependent. And I have underlined, and I'll box in so you guys can see the pink a little better, the 3 areas that are very highly hormone dependent. Is this part located in the medulla? Not all of it, right? Definitely not the DCT and not the collecting tubule. Those are both in the cortex. Portions of the collecting duct are up in the cortex, and portions of the collecting duct are down in the medulla, down in the pyramid, to be more specific. Does that make sense based on the anatomy of the system? Cool. So hormone dependent. Let's use knowledge you've already learned in the past. Again, this is all about making sure we're using the knowledge that we've learned. If I want to reabsorb more sodium, isn't there a hormone that we've learned that allows that? What hormone is all about sodium retention? Renin can trigger sodium retention, Heather, but it's doing it by activating another hormone. Great answer, though, because you know how I feel about renin. Melissa, you can text me that. We can talk about it again later. We're going to be talking about the micturition pathway probably tomorrow. So the answer is aldosterone. ADH is the other one, antidiuretic hormone. So not a lot of people writing this down. Good job, Tariq. It's okay, everybody else. It happens. The answer is aldosterone. 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 You gotta know this. You gotta know this. You gotta know this. You gotta know this. You must know aldosterone. End of the story. I will test you on aldosterone 1,000 times over. Aldosterone's all about sodium retention. So sodium reabsorption. And at the same time that it is reabsorbing sodium, it's allowing for potassium secretion. You guys remember that now? Is this ringing a bell? Awesome. Gotta know aldosterone. End of story. So the way aldosterone works is it's going to act on the DCT, the collecting tubule and the collecting duct, and it's going to upregulate transporters that allow for sodium reabsorption and potassium secretion. We'll deal with this more again later. So just for now, write it down so you have it in your brain. We'll make use of it. Now, what if we want water reabsorption? Heather. Aldosterone does not make you pee. Aldosterone is all about sodium reabsorption because now as we're reabsorbing more sodium, party's going back into the blood. What about water retention? What hormone is going to be involved in water retention, water reabsorption? Help me help you. Now we're dealing with ADH. Good. Antidiuretic hormone. This is about water retention or water reabsorption, same thing. And how is this going to work? Again, protein transporters. The DCT, collecting tubule, and collecting duct are normally impermeable to water. However, as long as ADH is in the system, we're going to now have protein transporters, aquaporins, aquaporin 2 to be more specific, that is going to be inserted into the cell membranes. And as it does, water can go to where that party's at. Sound good? We'll deal more with this later. We'll draw it out. We're going to actually draw out the tubule cells and apical surfaces and basal surfaces and where the transporters are found. But does this all make sense to y'all? Cool. So the way it works is PCT never changes. PCT, you have a nice pick. PCT is reabsorption of all sorts of materials, sodium and potassium and chloride and bicarbs and glucose, and the number of transporters here do not change much. There's not a lot of up and down regulation going on here in the PCT. Just going to happen. And there are aquaporins here. Therefore, as all of these ions and glucose get moved back into the blood, water gets to follow. Whereas down here, we don't have those protein transporters for sodium or any of the other ions, and glucose is totally not going to get reabsorbed here. But it is permeable to water. Therefore, water can move to where the party's at, which is in the blood. Why is there a party in the blood? Because the blood's flowing in the opposite direction. And just before this, in the blood flow, is just after this in the filtration system. So the ascending limb of the loop of Henle has these sodium 2-chloride potassium symporters, and all of these salts are getting moved into the blood here in the vasa recta. Sorry, kiddos. Party in the blood, water follows. End of the road is all hormone dependent. Hormone dependent. Don't tell me any reabsorption occurs here without the help of hormones. It just doesn't work unless we're talking about hydrogens. And then we'll talk more about hydrogens later because those intercalated cells are going to be involved in that function. All right. The last little property or process of the nephron here is the fact that not everything can be filtered. Some things are simply too big, too charged, and they just cannot be filtered, no matter how big the fenestrations are. But we still need to get rid of this stuff. So what are we going to do? We're going to use protein transporters, and we're going to move this back into the tube. This is called secretion. We're going to take protein transporters, move things back into the tube. This is called secretion. There is a small amount of secretion that still occurs in the loop of Henle, but nothing that I found good, solid data on, so I'm not going to teach you guys about that at all. Secretion. The primary secretion is going to be occurring up in the PCT and in the DCT, and a little bit of secretion over here into the collecting duct as well. Secretion is always an active process. It's always going to require ATP. We're always moving it via ATP from the blood back into that filtrate. And it's typically going to require a trigger, not always. And by a trigger, I mean like a hormone. Let's see here. What's being secreted? Things like, well, they are organic anions. So anything that is an organic anion, like if an amino acid or a lot of these other large organic anions. Let me try to. Most of the organic just basically means a carbon-based. So if we need to get rid of any of our fat chemistry, well, fat chemistry, of course, can just, it's non-polar, so it's just going to go through the cell membrane. But the large majority of our proteins, with the nitrogen bases and the carbon bases and the amino acid bases, we need to get rid of these. And then there's also larger metabolic waste properties, so like chemical reactions that are occurring that are creating metabolic waste that is carbon-based, organic anions, again, a large charged molecule that's organic with a carbon base. Those are the sorts of molecules that are going to get secreted. Some examples that the human body is making is something like creatinine, for instance. And you're, into exercise, so you know about creatinine probably. Creatinine, creatine kinase, storage of, yeah. So something like that would be a great example of something that is secreted. Yeah, creatine and creatinine as well, right? Creatine and creatinine are slightly different. Creatine is going to be a byproduct of. Okay, cool. So using this, of course, now at the end of the road, what we have is if it makes it into the minor calyx, this is all stuff that's going to become urine. So we say that it is excreted. So we can now take all of this information and write out that formula. Excretion equals what goes into the system minus what leaves the system plus the small amount that we put back into the system. That's it. Excretion equals filtration minus reabsorption plus secretion. Everybody have all this information down? Nice. All right, so let's go to the lecture itself. Just follow along with me, okay? Because this stuff you've all written down. So now we can follow along with, I don't know, the next 15 slides, no problem. I guess 13 slides, no problem. So 4 processes in the nephron are filtration, reabsorption, secretion, excretion. All filtration is going to be occurring between the glomerulus and the Bowman's capsule. Got the efferent arteriole in, efferent arteriole out. And this is going to allow for massive amounts of material to be filtered, 180 liters per day. 180 liters per day, that's going to be 100% of your filtrate volume. Not 100% of your blood volume, 100% of your filtrate volume. We'll talk more about the milliosmolarity in probably just a second, actually. Now, this filtrate enters the system, and that's it, because there's no more filtration that can occur anywhere else. So all of this volume gets added. What happens next? We reabsorb. Lots of different types of reabsorption that are occurring in the PCT. The thin descending limb of the loop of Henle, the thick ascending limb of the loop of Henle, the DCT, and the collecting duct, collecting tubule over here. We'll deal again with volumes and the milliosmolarity in, I don't know, another part of a lecture. I'm not going to skip it. It is important. Now, a little bit of secretions occurring through these other systems here in the DCT, in the PCT, DCT, and the collecting duct, just small amounts, really negligible in terms of measuring the amounts. We can do it, but we'll have to actually go through and learn about clearance, which we'll do later. End of the road is excretion. So look at the whole system as a whole. Whatever we filter, that's the large majority of the sum that goes into the system. We get rid of some and we add some, and whatever's left over is excretion. Pretty simple idea, I think. I think you guys are well advanced at this point. Where it starts getting more complicated has to do with volumes of blood, volumes of plasma versus volume of filtrate. And we already saw that in today's lecture a little bit earlier, right? There was a little bit of confusion of what's going on with the 10s and the 20s, 20%. Well, a lot of this has to do with what actually is getting filtered. And what I need you guys to try to remember is that only plasma gets filtered. Compositions of the blood, right? You got your hematocrit. You got your hematocrit. And you have your buffy coat, which is negligible, right? Those are the platelets and stuff like that. You're not going to be able to measure those, really. And everything that's left over is plasma. So let's just do a quick problem solve. Everybody pay attention to the words. I'm not going to write it down. If you have a patient and their hematocrit, let's do real simple. If their hematocrit is 50%, what percent volume of their blood is plasma? Nope. Try again, bud. And Ruju, don't take into account the buffy coat, because the buffy coat is negligible. Buffy coat is way less than 1%. Y'all, your blood is red blood cells and plasma. There are some white blood cells in there and platelets, but those things, they just don't take up a lot of volume. Yes, no, maybe so? So if 50% of your blood is red blood cells, then the other 50% of your blood is plasma. Work with me on this. If 50% of your blood is red blood cells, everything else is plasma. Because white blood cells and platelets are negligible in terms of total volume. Does that make sense? This will help you for your lab practical and your lab report, I promise, because you're going to have to do some math on that. Lots of math, actually. If your hematocrit is 45%, how much of your blood is plasma? There we go. If your hematocrit is 30%, I don't like it to be down there, that's anemia, but if your hematocrit is 30%, how much is plasma? All right, everybody on board now? Only your plasma can be filtered. That's it. It's the only portion of your blood can be filtered, because red blood cells are ginormous and they're charged. There's no way they're going to get filtered. If you have red blood cells in your urine, you got a problem, especially if it's because it was able to be filtered. If you get filtration of erythrocytes, that means you have complete renal failure at this point, most likely. You can get some blood due to urinary tract infection. So anywhere along the road, if you get a urinary tract infection, you can get red blood cells in your urine, no problem. But if it's coming from your glomerulus, if you're getting RBCs through here, there's a problem. 21-month-old daughter is trying to open up the front door and leave. Okay, so as that plasma volume enters, whatever the volume of plasma is going to be 100% of the plasma. So again, that could be a little confusing for students, but if 50% of your blood is plasma, 100% of the plasma volume is entering those kidneys. And of the plasma volume, it's 20% of that plasma volume that is going to get filtered. Therefore, 80% of the plasma volume goes right back into that efferent arteriole and out into the peritubular capillaries. Only 20% of your plasma volume is getting filtered. That's a big number though still, right? 20% of volume. If 20% of your blood is going into your kidneys in the next 60 seconds, well, 20% of your blood is about a liter of blood. And of that, about 50% is plasma, and of that, about 20% gets filtered. So that's the 180 liters per day getting filtered. It's a huge amount. You guys will not have to do any of that math that I just did, but I do want you guys to have a concept of what's going on there. Of that 20%, 180 liters per day, you're not peeing all that out, therefore the large majority of it's going to get reabsorbed. This is going to allow your blood volume and your blood pressure to hopefully be maintained. 2% gets filtered. Hold on. Huh? 20% gets filtered. %20 of your plasma gets filtered every minute. Are you talking about 20% of 20%? So you're talking about total volume, Tariq? Right, 20% of 20%. I actually have never really done the math on that, but which slide deck? This is just urinary too. You guys are overthinking the heck out of this. Stop. All we're trying to do is we're saying we got a big plasma volume that's entering, right? We got 100% of whatever your plasma is, that's 100% of 20% of your blood that's coming in. That is a large volume. Okay, that's like, 180 liters, so like 1,000 liters of plasma that's entering your kidneys every day. That's insane. Of that, about 20% is going to get filtered, which is 180 liters per day. Of that, the large majority it's going to get reabsorbed. It's got to go back into that plasma so that when we pee, we're only peeing out very small amounts. And the large majority of this pee, hopefully, is just things we don't need. But we always have to, resupply the system. It's just how it works. So if we're peeing out 1% of our blood volume, how much of your blood volume are you going to need to drink? You're going to have to put back in that 1%. Make sense? Yep, keep it simple, stupid. You got it. KISS. I always loved them. That was one my dad always told me. Cool. All right. Whew. A lot of work just to get there, but I hope you guys are with me on this. It's not that complicated. 20% of the plasma that goes through the kidneys is going to be filtered, plus or minus. This is called our filtration rate. More specifically, it's called our glomerular filtration rate. GFR. Can you guys all write that down for me? We're going to come back and do lots of math on that tomorrow. We're going to actually get to it today, and that's going to be kind of the end of where we're able to go through today. So this volume, this 20% of your plasma that gets filtered is called your GFR, which is an acronym that I gave you earlier this semester for zona. You guys remember those? Zona glomerulosa, zona fasciculata, and zona reticularis. So the whole idea of giving you that acronym in Unit 1 is to make it easier to remember the acronym now. So here you go. GFR means glomerular filtration rate. And that's what's going on here. Okay, this is a very important value. This value right here, 20% of the volume that gets filtered, that's homeostasis. So what if this goes up to 22%? Your body very well might not like that, right? If this volume gets too high, what's the body going to try to do to the amount of GFR? Increase, decrease, no change. And if this GFR drops to 15%, boom. What do we got going on? Increase, decrease, no change. Good, good. Making sense? What we're going to do is we're going to have to filter more, we're going to have to filter less, and there's going to be a lot of properties that we're going to walk through to try to understand how this system works. How this system works is related to this right down here. Excretion equals filtration minus reabsorption plus secretion. So how do we filter? How do we reabsorb? And how do we secrete? We already know the answer to reabsorption and secretion. How does reabsorption and secretion work in a human body, y'all? What do we require for reabsorption and secretion? Protein transporters, you got it. Filtration, we already have a background in filtration. This is going to be related to net filtration pressure. I know it's a little bit in your past, so you might not remember it completely, but net filtration pressure is all about the hydrostatic pressures and the colloid osmotic pressures. Do you guys remember those two words vaguely, or maybe even better than vaguely? It's from A&P2. We did all that in Unit 2. So we actually had a formula for that. So that's what we're going to be doing the rest of today and tomorrow and Thursday is we're going to be walking our way through how each one of these variables works. So for instance, filtration, what we're actually interested here in filtration, it's not just filtration. We're interested in glomerular filtration rate because the glomerular filtration rate is going to be what drives filtration. So depending on how much gets filtered over time, this is a rate, that's going to determine how much we excrete. If GFR goes up, what's going to happen to the total amount that gets filtered? Easy peasy lemon squeezy. Increase, decrease, no change. If GFR goes up, filtration goes up. And therefore, what should probably happen to the amount that we excrete? If we start having a higher GFR, we're going to excrete more, assuming everything else stays the same. Make sense? Well, glomerular filtration rate is a rate, just like the heart rate, just like the respiratory rate, it's a rate. And like heart, we talked about cardiac output being the primary function, which is mils per minute. And in the respiratory system, we talked about pulmonary ventilation, right? Which is mils per minute. Guess what GFR is? It's a rate. It's the homeostatic value of kidney function. It's going to be what you're measuring in real clinical scenarios. You're going to be looking at the patient's GFR to look at kidney function. Well, guess what? It's in mils per minute. It's a GFR. It's how many mils of your plasma are you filtering over time? Does that make sense? Now the variables are going to be different because we don't have, you know, a heart rate or a respiratory rate to be dealing with. So the variables that go into this, you're not going to have to mathematically figure out. And I just won't make you do it. But GFR, the primary variable that affects GFR is going to be net filtration pressure. And this is the one that you guys learned earlier this semester, where net filtration pressure is going to be equal to a hydrostatic pressure inside that system. This system's not a typical capillary, so we're not going to say hydrostatic pressure of the blood. We're going to say hydrostatic pressure of the glomerulus. So in this particular case, we're going to do a subset G. Okay, the hydrostatic pressure inside that glomerulus is going to decide how much pressure is being applied to the walls of this capillary. Does that make sense? All right, next. What do we have in our bloodstream that is the party of your bloodstream? Sorry if you can hear my kid screaming in the background. I'm sorry. It happens. Not just any particles. What is living in our bloodstream? That's the party, the primary party. That's a G, Heather, as in glomerulus, because it's the hydrostatic pressure inside the glomerulus. So what's inside of our blood? Everybody, please, I need your help on this. I got one person that's got it so far. Proteins, awesome. Proteins. What's the primary protein that's in our bloodstream? That's the party of your bloodstream. Andy's all over it. You guys can just copy her. Hopefully that is stuck in your head to some degree or the other. It's albumin, right? So we got a protein, and we're going to have a pressure related to that protein. So it's colloid osmotic, right? Colloid, as in a protein in solution, osmotic because colloid is the party, and these are pulling pressures. They're vacuum pressures. They're trying to draw water to it. So the more colloids in your plasma inside that glomerulus, the more it's opposing filtration. Does that make sense? Cool. What else do we have involved in this? Okay, what else do we have involved in this? Well, we have a Bowman's capsule now. We actually have a physical space that's filling up with filtrate. So we're getting more fluid here than we would in the interstitial space in the story. There's a lot more fluid here. So there's going to be a lot more hydrostatic pressure pushing back against the glomerulus this time. So if it's pushing back, then it's opposing filtration. So we're going to have hydrostatic pressure inside that Bowman's capsule. Bowman's capsule. That's going to be pushing back against that glomerulus. So it's opposing net filtration pressure. Does that make sense? Based on what we learned earlier this semester and based on your knowledge now? Cool. The last thing, this is the only value that should typically be 0 in this system. Should also be almost 0 in normal capillaries and normal tissues as well. But this is even more important, that it's as close to 0 as possible. Because as we're filtering materials, one of the major players that you do not want to filter are your proteins. We do not want proteins to be out here. Because if proteins are being filtered, that means that the filtration system is broken. Proteins are too large, they're too in charge, and they should not be filtered. But if proteins were to happen to get out here, we are going to pee those proteins out. If proteins were to happen to get out here, where's the party at now? The party's in that Bowman's capsule. Does that make sense? Therefore, this is promoting filtration. This would be colloid osmotic pressure, but it's in the Bowman capsule. This value should be what, though, everybody? Colloid osmotic pressure in the Bowman's capsule should be what? 0, 0, 0, 0. There are a few situations where you can increase the pressure in your glomerulus so high that you'll actually start to pee out albumins. One is like extreme exercise. If you over-exercise, it puts too much strain on your glomeruli, and that actually causes you to start to pee out albumins. You'll get albumins in your urine. Or a super high protein diet. Your body really just has no other way to get rid of all those extra proteins. So yeah, definitely a small problem there. So GFR is going to be, well, it's not equal. I shouldn't do that. It's proportional to NFP. It's not equal to NFP. It's proportional to NFP. It's proportional to NFP. So again, if any of these variables in our NFP start to change, it will impact your GFR. Let's put this into perspective. Again, I like doing these little, problems. So let's go with the patient that has, I don't know, liver failure that's affected how much albumin's in their blood. So they have some sort of hepatitis C. They are decreasing the amount of albumins. They're having hypoalbuminemia. Therefore, the colloid osmotic pressure inside the blood starts to drop. What's going to happen to their NFP? Increase, decrease, no change. Remember, we're taking smaller of negative. And if there's less proteins in the blood, there's less. Good. There's a bit of a lag going on where I'm at right now, so. But you got it. NFP starts to go up. And if NFP goes up, what's going to happen to that patient's GFR, potentially? And if filtration goes up, then they potentially have polyuria. It's just a possibility based on kidney physiology. There's a lot more going on in these patients. I hope you guys understand that. But based on just the renal physiology alone, they would have polyuria. They would have too much pee. Now, I want to go one step further. We're going to end up throwing in another value here. I don't want you guys to ignore the fact that it exists. There is another value that's going to be affecting GFR, and it's called the filtration coefficient. Coefficients in math are K's for whatever reason. Filtration coefficient, so a lowercase f. And we're going to be talking more about that tomorrow. Okay, just put that into your formula now. But it's easy to think about in terms of the physiology, because if the filtration coefficient goes down, it's directly proportional to GFR. What do we got? If the filtration coefficient decreases for whatever reason, we'll talk about the physiology of it, then GFR drops, and if GFR drops, then filtration drops, and if filtration drops, then excretion drops. Make sense? Awesome, you guys rock. You guys rock. One more idea I'm going to throw at you, and then we're going to actually call it quits, is that excretion is the same thing as saying urine volume. So UV, right? This is urine volume. Excretion is proportional to urine volume. So if we have a higher excretion, the urine volume is going up. Yes, no, maybe so? Vice versa. Then I want to remind you guys of a really important function that ties back to why the kidneys are so darn important. If you have an increase in urine volume, where's that coming from? Excretion came from filtration. Filtration came from your blood. So as urine volume goes up, what's going to happen to that patient's blood volume? Increase, decrease, no change. So blood volume is inversely proportional to urine volume. And I need you guys to hold on to that for the rest of the semester. Because if we have a patient with polyuria, mini, urea, urination, what's going to happen to their urine volume if they have polyuria? Boom, increase in that urine volume. Therefore, what happens to that patient's blood volume? You guys just did it. Blood volume drops. And what should we be thinking about when we're thinking about blood volume? What's the next step in your thought process? Maybe, maybe not, I don't know. BP, you got it, Andy. The next step in your process, I'm hoping you guys get here. You got to be thinking if a patient's blood volume changes, it will impact their mean arterial blood pressure. Yes, no, maybe so? Where we have cardiac output times TPR times blood volume. So now that blood volume drops, mean arterial pressure starts to drop. What two other values is the body going to try to kick in to compensate? If blood volume drops, we can't fix that. We have polyuria. We can't fix that. It's broken. So what two other variables can the body use to try to bring that blood pressure back up again? So now we have a patient with polyuria, hypertension, and tachycardia. Does that make sense? It's all about compensation. It's all about homeostasis, and that's going to be your guys' future in the medical world, is trying to think through these problems before they ever occur. Why am I diagnosing this patient with tachycardia? I don't know what's going on. Well, let's start thinking about some of the things that can cause tachycardia. Polyuria is one of them. So let's just check and ask them history. You know, you've been peeing a lot lately? Pretty simple thing to do, right? And then you can start looking into renal issues. Even though you are diagnosing them with a heart problem, maybe they don't have a heart problem. Maybe they actually have a renal problem. Make sense? I hope it does. I hope it does. All right, we're going to call it quits a little bit early here. I'm kind of at a point where if we're going to go to the next part of the slides, it's going to take a lot of time. So tomorrow, come prepared. Tomorrow, come prepared. We're going to be talking about the intrinsic and extrinsic controls of GFR. That's a pretty big deal. Intrinsic, we know is local. Extrinsic, we know is either hormones or nervous system. And you guys hopefully have a background in that now. That makes it a little bit more tangible. That's my hope. All right. Peace out, everybody. Text me if you have any questions. Yes, I am starting my drive here soon. Got about 6 hours of crazy. So yeah, you guys keep up the great work. This stuff's not easy, so it takes a lot of time and a lot of effort. But I know you guys all have that motivation to do it. So best of luck. Bye. Melissa, I just saw what you wrote. I never made it available yesterday, did I? I'll do